Hello, everyone. We published all these data and several papers came, around 60 papers came from our group about the epithelial barrier hypothesis and theory, and you can go through. And one of the important things for you to know is that there's comorbidities and multimorbidities have also increased during the time, and they are also linked to epithelial barrier diseases. So the epithelial barrier theory is still one of the most prevalent theory to discuss, and we are talking about increased prevalence after 1960s and 2000s. And when we select diseases, we can find now up to 40 diseases that fulfill all these four criteria. The second criteria is microbial dysbiosis with loss of commensals and colonization of opportunistic pathogens. Circulating microinflammation is a new term that we are introducing. And epithelial barrier defect and epithelitis. Epithelitis is also a new term that when we are exposed to environmental substances, toxic substances, epithelitis is the initiator of this disease. Here is a list of diseases. These are the ones that the affected tissues have epithelial barrier problem. You can see here atopic dermatitis, asthma, chronic rhinosinusitis, allergic rhinitis, eosinophilic esophagitis, inflammatory bowel, and celiac. And here is another group of diseases. These are autoimmune and metabolic diseases that are linked to gut barrier defect and microbial dysbiosis, diabetes, obesity, fatty liver, autoimmune hepatitis, liver cirrhosis, rheumatoid arthritis, multiple sclerosis, systemic lupus, and ankylosing spondylitis. The third group of diseases are chronic neuropsychiatric conditions. We don't have good models, but when you make biopsies on the right time, you can see that there's enteric inflammation, barrier defect, and microbial dysbiosis is seen. The group of diseases are here: autism spectrum, Parkinson's disease, Alzheimer's disease, stress-related psychiatric disorders, and chronic depression. And the prevalence of these diseases are also increasing nowadays. So we come now to the mechanisms. One of the most important mechanisms is a healthy interaction of the microbiome and epithelial cells is broken. And this homeostatic interaction has an agreement of several hundred million years with the mammalians and with the animals and also with the microbiome. So as humans, for several million years, we are carrying these microbes in our body. And these microbes have a certain job. They have to have digestion, nutrients they have to provide, and vitamins, metabolites, and they have to take a role in education of our immune system, and they have to help the differentiation of epithelial cells and immune system cells, and they have to help the maturation of the tissues. We know this from the germ-free mouse and normal mouse very well, and we can also understand that these are taking place currently. But what happened is that this agreement is broken since 1960s in humans, and the biodiversity of the microbiome and also the number of strains that we are carrying about 1 kilo of microbes in the gut and in our body in total, and there's around 4,000 strains, so which decrease very significantly. And this live together, co-evolve, select, and support reaction is not happening. The first thing that to start when we are exposed to this environmental substances is the epithelitis. And epithelitis is showing together with migration of the microbiome deeper, and also translocation of some of the microbiome. At the same time, this irritated epithelium starts to make interleukin-1, interleukin-25, interleukin-33, and TSLP. So TSLP is available now as a treatment modality for asthma. And this increased alarmin production invites the immune system as close as possible, and then inflammation starts. This inflammation is together with neuroimmune interactions, and the activation thresholds of mast cells, dendritic cells, are also regulated. Now remember the expulsion response. What is expulsed? And the second thing what happens is the expulsion response. So what is expulsed? It is not the parasite larva, but what is expulsed is the microbes that are trying to go in between, touching the epithelial stem cells or translocating below the epithelial cells should be expulsed. To expulse these microbes, we are showing an immune response, and this immune response is a type 2 response in most cases, and together with it, we have IgE, eosinophilia, T helper 2 cells, ILC2, IL4, IL5, IL13. And at the same time, we have colonization of opportunistic pathogens, mainly for the respiratory area. We are talking about Staph aureus, Moraxella, Haemophilus, and Pneumococcal. And the biodiversity decreases for 3 reasons. The first one is colonizing opportunistic pathogens. The second one is immune response to commensals. And the third one is direct toxicity of environmental substances to commensals. So all of them are taking place. And then there is an autoimmune reaction that's taking place at the same time. And immune response to commensals, mainly to staph aureus, is becoming very dominant in these patients. So circulating microinflammation is an important concept that you are now learning, maybe hearing for the first time, but this is quite important. Now, if you focus on this section of the lung or on the section of the gut, we see that the epithelial cells are not touching each other, and the microbes are passing through the epithelium, and the cells are activated, and we have now clear evidence that T helper 1 cells coming from the lung and going to brain can cause multiple sclerosis, and T helper 17 cells and T helper 1 cells can go to joint and make rheumatoid arthritis. And there is several evidence for this, like CLA positive skin homing cells from the gut. This was the PhD thesis of Mübeccel, and Donald Deering worked on this area. Impaired gut epithelial barrier before diabetes onset, joint migrating T helper 17 cells, brain migrating T helper 17 cells, they were all reported. Now here's actually a conclusion slide. I would like to discuss with you the epithelial barriers are damaged, and for genetical reasons and exposure to environmental agents, this damage occurs, and colonization of opportunistic pathogens is taking place, translocation of microbiota to subepithelial areas, and immune response to commensals and opportunistic pathogens, and loss of commensals, microbial dysbiosis, and decreased biodiversity is taking place. Then the inflammation of the epithelium and chronicity, that is a chronic expulsion response, is taking place, and defective barrier healing capacity is happening because of epigenetic reasons, so that this barrier can never become a full healthy barrier. So this ends up with 2 billion allergic, autoimmune, and neurodegenerative patients. So if we summarize, we have direct toxicity to epithelium, epithelitis, activation of the epithelitis, release of alarmins, expulsion response, inflammation, and to deep translocated microbiota is taking place. Migration of inflammatory cells to distant organs, so gut inflammation causes chronic hepatitis or brain disease or arthritis, and we have epigenetic regulation. Very strong epigenetic mechanisms play a role in defective barrier healing capacity.